Hello all, welcome to my presentation. I am MD Niyazul Islam Khan, a graduate student of Electrical and Computer Engineering Department of University of New Hampshire, Durham. The title of my presentation is Electrochemical APTA Sensor based on carbon nanotube modified screen printed electrodes for lysozyme detection. Lysozyme is a protein. So the question comes to our mind that why we need to detect lysozyme. There are several reasons. First of all, lysozyme is an antimicrobial protein. That means the presence of lysozyme uh, can kill some microorganisms that are harmful for our bodies. It has clear structure and low molecular weight. They are used in protein research, medical treatment as biomarkers and food industry. Biomarkers are those biological substances um, that are used to diagnose different diseases, for example, cancer or Alzheimer's, etc., can be detected with lysozyme biomarker. So lysozyme detection is very important to us. There are different detection techniques for detecting lysozyme. For example, electrophoresis is one of them, liquid chromatography, enzyme-linked immunosorbent assay, or ELISA in short. But these detection techniques are not free from disadvantages. They have several disadvantages, like they are expensive, they are not portable, they are also complex, also requires protein modification that is very tedious. So we need to overcome these disadvantages. So of the paper is to design and implement a low-cost and portable molecular recognition element to detect lysozyme with good sensing performance. So this to achieve the goal, our hypothesis is to use aptamer as the recognition element. If we can use aptamer as the recognition element, it will reduce the cost because aptamers are very cheap. Use disposable screen printed electrodes to increase the portability. Use carbon nanotube to immobilize the aptamer on the screen printed electrodes. This is a figure of a screen printed electrodes. You can see reference electrode, counter electrode, and working electrode. The working electrode will be modified with aptamers, and this electrode can be used for electrochemical impedance spectroscopy measurements. What is an aptamer? Aptamers are synthetic, short, single-stranded oligonucleotides. They are more stable over a wide range of pH, temperature, and salt condition than natural antibodies or enzymes, etc. They have high affinity and specificity with respect to target analyte, low cost, and have low molecular weight. Carbon nanotube. Carbon nanotubes are used to immobilize the aptamers on the screen printed electrodes. They have versatile advantages, like they are highly conducting and they have increased surface to volume ratio that will increase the density of aptamer on the screen printed electrodes. So our sensitivity of the aptamer biosensor will be increased, can be increased by the carbon nanotubes. This is a figure of a carbon nanotube. So how we will test our hypothesis? We will do several steps. First of all, we will modify the screen printed electrodes with CNT and DNA ink. In the ink, the CNT will be wrapped around by the DNS. You can see the electrode here, and you can see lots of DNA wrapped CNTs, and this is the enlarged of a CNT. You can see that the DNA wraps around the CNT, and when we do the electrochemical impedance spectroscope measurement, the charge transfer from the redox probe to the electrode is blocked. And when we incubate lysozyme, the one part of the DNA will unbind the CNT, and it will bind to the lysozyme. So this part of the CNT will be open, and the charge transfer from the redox probe to the electrode will be more easier. So this electrochemical impedance measurement is illustrated in this graph. Uh, we expect a graph like this, and there are two curves. They are called Nyquist curves. The radius of the Nyquist curve indicates the charge transfer resistance. So the red curve is the curve before lysozyme incubation, and the other curve is after lysozyme incubation. The blue curve has a lower charge transfer resistance. It is clear from the figure, and so it can be said that lysozyme incubation reduces the charge transfer, uh, or in other words, it will enhance the charge transfer from the redox probe to the electrode. So we expect such a curve from our biosensor. Background: there are several works related to this. Few people did different ways. For example, Sen and co-workers investigated an electrochemical lysozyme aptamer based on thiol gold binding. They use gold nanoparticles to immobilize the DNA aptamers on the electrode. RDM and co-workers use magnetic particles for immobilizing the aptamers on the electrode. They use two cognate proteins like lysozyme and thrombin as the analytes. And the electrochemical detection was facilitated by using disposable graphite electrodes. Rohrberg and co-workers has designed an electrochemical lysozyme aptasensor based on amine coupling between the carboxyl group modified carbon nanotube and amino-linked aptamer. These are the references I used for my paper. Any questions you have, you can ask to me. I'll be happy to answer the questions. Thank you very much.